Hi, I'm teacher Yumni. Okay, today I would like to continue lessons with my students. And today topic is 6.2 light dependent reactions. Okay, before we start, I would like to share this first. Okay, so photosynthesis, as you know, that occurs in two stages, which are light dependent reactions, which take place on the Telecoil membranes, and light independent reactions that take place in the stroma of the chloroplast. And today topic is light dependent reactions. Okay, light dependent reactions consist of non-cyclic photophosphorylation and cyclic photophosphorylation. Okay, then if you can see on the screen now, in the previous lesson, you have already learned the components of a photosystem. Now, can you identify these components? Chlorophyll A, Telecoil membrane, carotenoid, chlorophyll B, and you got photosystem 1 P700 and photosystem 2 P680. Okay, now let's recall back what is a photosystem. So photosystem, it is the light harvesting unit of photosynthesis located in the Telecoil membrane of the chloroplast. And now I click play button and I want you to observe what happens when light energy strikes on the photosystem. As you can see here, light energy is transferred from one molecule to another till it reaches the reaction center, chlorophyll A. Okay, then so light reaction can proceed in two pathways, as I mentioned, cyclic photophosphorylation and non-cyclic photophosphorylation. Okay, let's start with the non-cyclic photophosphorylation. Okay, non-cyclic photophosphorylation involve two types of photosystem, which are photosystem 2 and photosystem 1. Okay, they name the P700 as photosystem 1 because this photosystem discovered first. Okay, then non-cyclic photophosphorylation got two-stage process. Okay, I click on step 1 in the stage 1 involving of a photosystem 2. All right, then let's see what happens. Okay, as you can see here, light strikes on the photosystem 2 and the electrons in photosystem 2 or P680 are excited to high energy level. Then what happens to the excited electrons? They leave the photosystem 2 and transported as high energy level electrons to the primary acceptor of the photosystem 2 of or photosystem 680, P680. Okay, now what happens to P photosystem 2? An electron hole is formed in P680. Okay, then what happened on the next? Okay, now you can see water molecule involved now. All right, here. What happens to the water molecule here? Okay, here you can see that water molecule split into hydrogen ions and oxygen molecule releasing two electrons. So two electrons were released during this photolysis. Okay, what happens to the electrons released from the water molecule? So the electrons move to P680 to fill up the holes. And why does this happen? As I mentioned, to fill up the electron hole. Okay, so settle then. So this reaction we said photolysis. What is photolysis? Photolysis is splitting of water molecule in the presence of light energy to release molecular oxygen. So here, photodissociation of water also said occurs. Okay, two water molecules are complex by the complex of photosystem 2, the water molecules are split, electrons are removed from water to replenish the hole in the reaction in the photosystem 2. And then hydrogen ions or protons are generated and released into the telecoil space. Okay, now we move on to the step 3 and let's see what happens. Okay, from the primary acceptor, electron travel through the electron transport chain. If you can see here, excited electron transported to the cytochrome complex through the electron carrier plastoquinone. All right. Then what happens when the electrons reach the cytochrome complex? As you can observe here, the electrons lose some energy in the form of ATP because previously electrons are high energy level, and so when they travel down the carriers, the electrons lose that energy in the form of ATP. So how is ATP produced? Here ATP is produced from ADP and inorganic phosphate pi group by utilizing the energy released during the previous reaction. So what is this reaction called now? Photophosphorylation, as you know in respiration. Okay, then let's move to the next step. Okay, what you can observe here? Okay, you can observe electrons. Okay, so this is stage, another stage in the non-cyclic photophosphorylation. Now electrons go to P700 of photosystem 1. Do you think that electrons return to P680 of photosystem 2? Of course, no, because let's observe why. Because now electrons transported to the primary acceptor of P700. They are not returned back to the photosystem 2, but instead they are transported to primary acceptor of P700. Okay, so now step 6, from the primary acceptor in the photosystem 1, you can see electrons move to NADP plus. NADP plus in the presence of NADP plus enzyme reductase. Now, if you can see, NADP plus is reduced to NADPH. Okay, this is due. Sorry. So this NADP plus act as a last electron acceptor in the non-cyclic photophosphorylation. Okay, now where does the hydrogen ion comes from? It comes from the splitting up of water molecule during photolysis previously at the previous stage. So this reaction called reduction because NADP plus reducted to NADPH. Okay, so now let's see overall of non-cyclic photophosphorylation. Okay, the electrons from photosystem 2 or P680 are transported through the electron transport chain to P700, then finally reduces NADP plus to form NADPH. So during this process, ATP is also produced. This is called a non-cyclic photophosphorylation. Okay. Okay, so let's conclude this. What is the net energy gain during non-cyclic photophosphorylation? If you can see, it is one molecule of ATP and one molecule of NADPH. Okay, so from previous explanation, what is energy gain during both cyclic and non-cyclic photophosphorylation? So one molecule of ATP from cyclic photophosphorylation and one molecule of NADPH from non-cyclic photophosphorylation. Shoots here. Okay, then comes the protons and electrons fill up the hole. So the electrons travel through the electron transport chain, goes to the photosystem 1 and goes to the primary acceptor. And via ferredoxin, they reduce the NADP plus to NADPH. All right, so one molecule of ATP from cyclic photophosphorylation and one molecule of NADPH from non-cyclic photophosphorylation. And another one molecule of ATP here. Okay, so the conclusion for both like reaction is cyclic photophosphorylation occurs in cyclic manner where only P700 is involved and one molecule of, okay, from here, okay, to cytochrome complex. Okay, while in photosystem 1, cyclic photophosphorylation involve this red arrow, okay? Electrons excited, accept by primary acceptor, travels down to cytochrome complex, okay, via ferredoxin and via plastocyanin and return back to photosystem 1 at the cytochrome complex. Cyclic photophosphorylation release another ATP. Okay, now cyclic photophosphorylation, okay, uh, occurs in a cyclic manner where only P700 is involved and one molecule of ATP is produced. In non-cyclic photophosphorylation, okay, please observe the blue arrow here. Both photosystem 1 and photosystem 2 are involved in producing one molecule of ATP and one molecule of NADPH. All right, and it does not occur in a cyclic manner. Okay, so this is the comparison between cyclic photophosphorylation and non-cyclic photophosphorylation. If you can see here again, non-cyclic follow the blue arrow, okay, until the electrons accept by NADP plus with the presence of NADP plus reductase enzyme and reduce to the NADPH. While in the photosystem 1 is a cyclic, it's a cyclic phosphorylation. The red arrow, when the photon strikes the photosystem 1, the excited electrons accept by primary acceptor and transfers to the electron carrier ferredoxin until reach cytochrome complex. And at the cytochrome complex, they will travel down to the photosystem 1 again. So the energy release trapped in the form of ATP via photophosphorylation. Okay, so I already cover for cyclic photophosphorylation involving a photosystem 1 and I already cover non-cyclic photophosphorylation that include both photosystem 1 and photosystem 2. Then energized electrons are released from the excited chlorophyll A photosystem 2, P680 is picked up by the primary acceptor. The electron hole that is formed in P680 due to the transport of the excited electrons is filled up by the electrons produced during photolysis. The electrons then transported to cytochrome complex through the electron carrier plastoquinone, during which its energy is trapped to form ATP. Okay, then because of the low energy level, electrons from the cytochrome complex are transported to P700 through plastocyanin. So the electrons from P700 transported to NADP plus for reduction via primary acceptor and ferrodoxin. So this reduction is catalyzed by enzyme and ADP plus reductase. The net energy gain during non-cyclic photophosphorylation is one molecule of ATP and one molecule of NADPH. Okay, while in the photosystem 1 cyclic photophosphorylation, okay, cyclic photophosphorylation, the generation of ATP by by cyclic electron flow. Okay, let's look at the this phenomena. What happens when the P700 photosystem in the cyclic photophosphorylation? What happens to P700 when light strikes? So the same thing goes to the photosystem. Electrons in the photosystems are excited to a higher energy level. And then what happens to the excited electrons again is the same. They will transported to the primary acceptor. So let's look furthermore. Okay, if you can see here, where do the excited electron go? Okay, as same in the non-cyclic, the excited electrons are transported to cytochrome complex through the electron carrier paradoxine. So when the electrons reach the cytochrome complex, energy is released in the form of ATP, and the same thing, ATP is produced from ADP and inorganic phosphate pi group by utilizing the energy released during the previous reaction. And then how is ATP, and then now the high energy level electron is reduced to low energy level because their energy trapped for the formation of ATP. And this process is same
Okay, I found this on the
photophosphorylation.
web for please during the previous reaction.
Okay. Okay, so now okay, so in step 3, from the cytochrome complex, the electrons lose their energy and return back to photosystem 1 P700 via plastocyanin. So overall of the cyclic photophosphorylation start from when the light strikes the photosystem 1 and the electron from P700 is transported through an electron transport chain in a cyclic pathway to return back to photosystem 1 P700. Hence, it is called cyclic photophosphorylation. During this process, ATP is produced. So, in this cyclic photophosphorylation, only ATP is produced. Okay, then all the electron carriers involved in cyclic photophosphorylation are ferrodoxin, cytochrome complex, and plastocyanin. So if you can conclude from cyclic photophosphorylation, light reaction takes place in the photosystem present in the telecoil membrane. So light reaction occurs at the telecoil membrane because at the membrane, they can transport the proton, pump the protons uh, into the stroma, or from the stroma into the telecoil to create a higher concentration gradient via concept chemiosmosis so that ATP can be generated. So in cyclic photophosphorylation, only photosystem 1 is involved, while the cyclic photophosphorylation and non-cyclic photophosphorylation, both photosystem is involved. Then the energized electrons are released from the excited chlorophyll A, P700, is picked up by a primary acceptor. The excited electrons pass through electron transport chain and return to P700 by releasing energy, which is used to produce ATP from ADP and P phosphate inorganic. Okay, so that's all for cyclic and non-cyclic light reaction. Okay, what else? Okay, about chemiosmosis in chloroplast. Okay, energy is released as electrons flow through the electron carriers. Okay, now mm, we already come to this conclusion comparison. I already summarized in the another slides. Okay, this is all the things that I already told. Okay, so now I conclude on another slides. Okay, here. Okay, here, as you can see here, I like photosynthesis. You know why? Because photosynthesis give us two things that important in our life, which are oxygen and sugar. Right? Sugar or food. So water involved in the light reactions to produce energy in the form of ATP and NADPH that will shuttle in the Kelvin cycle. And then Kelvin cycle with the carbon dioxide will produce foods. Okay, so Kelvin cycle need ATP from the light reaction and NADPH from the light reaction together with the carbon fixation by means to produce foods or sugar. Okay, so if you can see previously, it starts with the photosystem too, so that easy for you to understand about light reaction. Okay, when the light strikes the photosystem in the photosystem 2, P680 so chlorophyll A at the P680 will the electrons in the chlorophyll A becomes excited and accept by the primary acceptor, and with the help a split of water, with the help of water molecules split, contribute electrons to fill up the hole in the photosystem 2, so that the excited electron will takes up by the primary acceptor and transfer to plastoquinone until it reaches cytochrome complex and release the energy. And then the electrons travel to the photosystem 1, then accepted by primary acceptor and travel through ferrodoxin and electrons accepted by NADP plus with the help of enzyme to reduce NADP plus become NADPH. While in the photosystem 1, it's a cycle. It's a short circuit to produce ATP. It just strike the photosystem 1, chlorophyll A in the photosystem 1 become excited and reach the primary acceptor, and after that they go to the ferrodoxin, ferrodoxin so that it will reach cytochrome complex, again produce ATP, and via plastocyanin it returns back to P700 of photosystem 2. Okay, so cyclic electron cycle and non-cyclic electron cycle. So if you can see here, this is cyclic. The color one is cyclic photosystem 1, as I mentioned over and over. Okay, now this is it. Comparison of electron transport. If you can observe in the non-cyclic electron source, electron source is a water, but in cyclic no electron source. And in non-cyclic oxygen release, yes, because splitting of the water molecule, while in cyclic no oxygen release. And non-cyclic terminal electron acceptor is a NADP plus, while cyclic there is no terminal electron acceptor. And non-cyclic terminal electron acceptor is the last electron acceptor. Right. So non-cyclic energy is temporarily captured in the form of ATP by chemiosmosis. Same thing goes to cyclic. Okay, here is the most differentiation. Non-cyclic photophosphorylation requires both photosystem, while cyclic only photosystem 1 or P700 involved. Okay, now talking about chemiosmosis, what do you know about chemiosmosis? Do you still remember about respiration previously? When the motor generated, okay, I said motor, no. Like ATP synthase, enzyme ATP synthase, acting like a motor rotation. When they rotate, they pump the protons in. All right. So once the net force movement of proton will generate ATP, right? So pigment and electron acceptor of light dependent reaction embedded in telecoil membrane. So chemiosmosis need a membrane, okay, same as a respiration. So energy from electron traveling through chain of acceptor used to pump protons from the stroma across telecoil membrane into telecoil lumen. Because right, so stroma has a lower hydrogen concentration gradient, while telecoil lumen has a higher concentration hydrogen. So proton accumulate in telecoil lumen. So as I said, it becomes higher concentration gradient as water is split during non-cyclic electron transport. So water is split in the telecoil lumen. So during non-cyclic electron transport makes the proton accumulated. So telecoil membrane are refused. Protons diffuse through a membrane except through certain channels formed by enzyme ATP synthase motor rotation. Okay, so it transmembrane protein free energy decrease as a consequence of and increases in entropy when protons diffuse through ATP synthase complex. So every four, okay, so from exergonic process, diffusion occur down concentration gradient to endogonic process of phosphorylation of ADP to form ATP and released into stroma. So every four protons move through ATP synthase, one molecule ATP is synthesized. So chemiosmosis, phosphorylation of ADP is coupled to diffusion down a proton gradient. Okay, how phosphorylation ADP is coupled to diffusion down a proton gradient? Okay, before we go further, let's look at here. Okay, so the hydrogen concentration in the telecoil space is higher concentration, so it will diffuse into the stroma lower concentration gradient. So at the stroma, ADP is converted to ATP, right? So here in both mitochondria organelles, Chloroplast organelle, electron transport chain pump protons across a membrane from a region of low hydrogen concentrations. Okay, from the low hydrogen concentration, hydrogen will pump into the higher concentration via electron transport chains. Okay, you got this? Okay, so electron transport chain pumps proton across a membrane from a region of low hydrogen to higher hydrogen concentrations. Then the protons diffuse back across the membrane through ATP synthase. So during this diffusing back across the membrane through ATP synthase, it drives the synthesis of ATP. Okay. So if you want to compare chemiosmosis in mitochondria and in chloroplast here, high energy electrons drop down the transport chain extracted from organic molecule sugars which are thus oxidized. While in chloroplast, the source of electrons are water. Chloroplasts do not need molecules from food to make ATP. Their photosystems capture light energy and use it to drive the electrons from water to the electron transport chain. Then mitochondria use chemiosmosis to transfer chemical energy from food molecules to ATP, whereas chloroplasts transform light energy into chemical energy in ATP. Okay, so it is about chemiosmosis. Now I want to wrap up this topic. Okay, so whatever things I mentioned, okay, previously. Okay, sorry. Okay, so here the diagram of the shows a current model for the organization of the telecoil membrane. The gold arrows, all right, track the linear electron flow as electrons pass from carrier to carrier in redox reactions. Hydrogen ions removed from the stroma. Okay, so this is stroma. Okay, hydrogen ions first enter the stroma via electron transport chains. All right, and then after that, they are removed. Okay, they are removed from the stroma and then deposited. Sorry, here through electron transport chain. Here, if you can see, hydrogens removed from the stroma and then deposited into telecoil space here. Okay. Okay. Now, here, telecoil space storing energy, store energy as a proton move force via hydrogen gradient concentrations. So at least, okay, I will show you steps in the light reaction contribute to the proton gradient. Okay, first, water. Okay, if you can see here, water is split by photosystem 2. Okay, water split by the photosystem 2 on the side of the membrane facing the telecoil space. Then plastoquinone, a mobile carrier, transfer electrons to the cytochrome complex. 4 protons are transported, okay, across the membrane into the telecoil space. Okay, the driving 4 protons, okay, translocated into the telecoil space. Then as plastoquinone, sorry, and then a hydrogen ions is removed, okay, from the stroma when it is taken up by, okay, sorry, here, another two. This is two protons pumps into the telecoil space and another two protons, okay, removed from the stroma when it's taken up by an ADP plus. Notice in this figure, hydrogen ions are being pumped. Hydrogen ions are being pumped out from the stroma into the telecoil space. From here to the telecoil space. Okay, then the diffusion of hydrogens, from the telecoil space back to the stroma along the hydrogen concentration gradient powers the ATP synthase. Okay, because the higher concentration gradient in the telecoil space will power the ATP synthase, and this light-driven reaction store chemical energy in NADPH and ATP. Okay, and NADPH and ATP together will shuttle the energy to the carbohydrate producing Calvin cycle. So total 4 hydrogen ions here through the electron transport chain and 2 hydrogen or 2 proton, 2 hydrogen ions in the stroma in the reduction of NADP plus into NADPH. Okay, so the same thing goes here. Okay, so 2 hydrogen ions, photon, will enter through the through the electron transport chain, and while in the reduction of NADP plus into NADPH, another 2 hydrogen ions. All right. So the higher concentration gradient in the telecoil lumen will drive the ATP synthase to produce ATP. And comparison between photosynthesis and aerobic respiration will conclude my this subunit. Sorry. Okay, so if you look at here, photosynthesis and aerobic respiration. Okay, photosynthesis need carbon dioxide and water, while aerobic respiration need sugar to being oxidized. So end product of photosynthesis are sugar and oxygen, while aerobic respiration end products are carbon dioxide and water. So which cell provides photosynthesis? Cell that contain chloroplast, while aerobic respiration, every actively metabolizing cell will respire. So sites involved in photosynthesis is a chloroplast, while aerobic respiration, they need mitochondria and cytosol for glycolysis. So ATP production via process photophosphorylation because they need light, and aerobic respiration ATP production via substrate level phosphorylation and oxidation phosphorylation. So electron transfer compound in photosynthesis is NADP plus reduced to form NADPH, while in aerobic NAD plus reduced to form NADH. Now location electron transport at the photosynthesis, electron transport at the telecoil membrane, while aerobic respiration, mitochondria in a membrane. So source electron for electron transport chain via non-cyclic water photolysis yield electron, protons, and oxygen, while in the aerobic respiration, source electron for ETC is a immediate source NADH and FADH2, and ultimate source, of course, glucose. Terminal electron acceptor, the last electron acceptor is in the non-cyclic NADP plus reduced to form NADPH, while in aerobic respiration, oxygen reduced to form water. All right, so that's all for lesson today. Okay, so I will stop my lesson now. I hope you can understand and try to do some revision and do exercise that I already gave to all of you. So, see you on the next lesson. Bye. Take care.